ATP synthase. And this is the enzyme that makes ATP. So essentially what happens is all of those hydrogen ions that are building up in the intermembrane space are going to flow through the floodgate that is ATP synthase. And as they flow through ATP synthase, they actually do spin a little turbine that's in that enzyme, and that gives off enough energy for the enzyme to make ATP. So here's what this looks like. This is our inner membrane of our mitochondria. So this is the matrix. And this is the intermembrane space. And what we've done with the electron transport chain is we've put a lot of hydrogen ions up here. There are tons of them up here, and there are not very many of them down here. So ATP synthase, which is this molecule here, is going to be the floodgate. All these hydrogen ions that are in the intermembrane space are going to flow down through ATP synthase, and that's going to power the production of ATP. And it powers enough that we get tons of ATP. Remember, 26 to 28 molecules of ATP. So here's this whole process together. Our NADH is going to deliver electrons to complex 1. As those electrons fall down this electron transport chain, they're going to power the transport of hydrogen ions into the intermembrane space. Eventually, we're going to make water. We're going to do the same thing with FADH2. It's going to go to complex 2, not complex 1, but its electrons are also going to flow down this electron transport chain. As they do that, they give off enough energy that we can pump hydrogen ions into the intermembrane space. Now, all these hydrogen ions are going to flow back down through ATP synthase, and as they do that, they power the production of ATP. And specifically, they make 26 to 28 molecules of ATP. So how many molecules of ATP did we make from glycolysis, pyruvate oxidation, and the citric acid cycle? From glycolysis, we made 2. From pyruvate oxidation, we made none. And from the citric acid cycle, we made 4. So if you combine those 4 that we made starting with glycolysis, we've made a total of 30 to 32 molecules of ATP in this process, and that is a lot. When you look at this picture on this slide, it looks like all of these molecules are like all next to each other and they're connected to each other in some way. They aren't. Remember what you know about the plasma membrane. The proteins are floating in the plasma membrane, so they are not linked to each other in any way. And in fact, if you think about it, there could be another protein complex 3 over here, and NADH's electrons could flow that way too. We're just looking at one of a series of complexes in the mitochondrial inner membrane. Keeping in mind that this membrane surrounds the entire inside of the mitochondria. There's a lot of surface area there. So here's just another picture, because one thing that students tend to get confused about, or a misconception, is that's why we have all these folds in the inner membrane, because that increases the surface area, and there can be protein complexes all through this. 1, 2, 3, 4. They can be everywhere. And the ATP synthase doesn't have to be right next door. And again, that can be a hard concept to understand, but what you need to remember is that we're building up a hydrogen ion concentration in this inner membrane space. And this inner membrane space is connected all the way around the mitochondria. So anywhere there's an ATP synthase, there could be one right here. And hydrogen ions would flow down through this. There's one here, so the hydrogen ions are flowing down through there. There could be another one here, and hydrogen ions flow down through there. So just keep in mind that this is, I'm going to say it's a lot of space. I know it's a tiny microscopic organelle, but for that organelle, that's a lot of space. And so we have protein complexes embedded all throughout the mitochondria.